And it is my great pleasure to introduce my friend and colleague, Dr. Carrie Tribble. She is an assistant professor and curator of the herbarium at the Burke Museum, University of Washington. She received her PhD in integrated biology at UC Berkeley in 2020. She was then a postdoctoral fellow and collections manager at the University of Hawaii, and now she has joined the University of Washington. Um, Carrie's research uses very sophisticated statistical phylogenetic methods and integrative systematics to model the evolution of complex and understudied natural phenomena. She works on diverse groups of plants that go from monocots to ferns and in multiple regions, ranging from South America, Hawaii, and more recently, the Pacific Northwest. She's been recognized for her scholarship by the NSF and Fulbright program through graduate and postdoctoral awards. So today, she's going to give us this talk on From the High Andes to the Pacific Northwest, Evolution and Biogeography of Geophytic Plants. So thank you so much for joining us, Carrie, and take it away.
Fantastic. All right, everyone can hear me, see the screen. We're good to go. All right. Thanks so much for the introduction, Ishel, and thank you to all of you for being here. This is a great pleasure to virtually return to my academic home in California, tell you a bit about some of the work that I've done over the past few years or so, and give you a little bit of a preview, perhaps, into what I hope to do with my research moving forward now that I have landed in my new home at the University of Washington. So, let's see, there we go. So I am a tropical plant evolutionary biologist. My motivation for my research comes from observing the variety and diversity of the natural world, the trying to understand the processes that have shaped that diversity. And I do this work under the umbrella of phylogenetic comparative methods. And this is a field of study that allows us to test macroevolutionary hypotheses about how any organism has evolved over long time periods, millions of years, using statistical models of evolutionary processes. One of the things that I love most about phylogenetic comparative methods is that it is a field that leverages evolution, botany, genomics, mathematics, statistics, and computer science, and brings in elements from all of those different disciplines into a single unified framework, the phylogeny, to understand and test hypotheses about how evolution has occurred. Some of the questions that I'm most interested in my research are how do species spread across landscapes and through time? How do organisms evolve in response to large-scale disturbance and change? And how can we document and preserve biodiversity in the face of ongoing crises such as anthropogenic climate change, land use change, and so on? And like Ishel said during the introduction, I am interested in these types of questions in various regions of the world. So one of the regions where I've spent the most time working is in the tropical Andes, and I'm going to spend a fair amount of time today talking about some of my work in the tropical Andean biodiversity hotspot. This is a photo from a collecting trip that I did in Peru in 2022. Also the Hawaiian Islands. I spent 4 years as a postdoc in Hawaii, and I'm originally from there. So the Hawaiian biota has a very special place in my heart, and is a region that I am deeply interested in. It has encouraged me to think about some of the conservation implications of the work that I do and the work that we all do as botanists. And most recently, the Pacific Northwest. I started my assistant professor and curator position here at the University of Washington in September of 2024. So it's been almost a year that I've lived here. And at the end of my talk today, I'm going to come back with really a couple of questions that have begun to emerge in my time here. Um, most of my research has focused on geophytic plants. Geophytes are plants that are herbaceous and they resprout from below ground buds. Geophytes have evolved multiple times independently across the vascular plant tree of life. So on the left side of your screen, there's a phylogeny showing images and a couple of examples of different geophytes in diverse clades across the angiosperms, as well as into some of the pteridophytes, ferns, and lycophytes. So there's a lot of taxonomic and evolutionary diversity in this clade. There's also not only just taxonomic diversity, there's also a lot of morphological diversity because geophytes generally have underground storage organs. So these are structures that the plants produce that serve as a place for the plant to store sugars and water. And those storage organs then fuel regrowth of the plants following seasonal periodic dieback. So these storage organs can be modifications of various parts of the sort of typical plant body plan. One classic example is the onion, which is a bulb. Bulbs are modified leaves, or the modification of the sheathing leaf base. But there's also underground storage organs that are modifications of stems, such as corms and rhizomes. I'm going to skip over some of this, but the sort of classification of these structures can get a little convoluted. Um, and then modifications of roots as well. In different forms, you can have roots that are expanded horizontally along the length of their axis into an elongate root tuber, or just at the tip of the root into more of a rotund root tuber. And so these differences in both sort of the tissue origin of the storage organ and the gross morphology of the storage organ have not really been delved into in terms of how those differences might relate to the overall ecology of these plants and whether there is any significance or if these are all sort of different but equal paths to being an underground storage organ and serving that role for a geophytic organism, a geophytic plant. Um, most geophytes we think are these structures, underground storage organs, are adaptations to seasonal climates. So one example of that would be understories of deciduous woodlands, where geophytic plants are often these spring ephemerals that will sprout long before the overstory has a chance to fully flush and leaf out. Take advantage of that period of time in order to photosynthesize, reproduce, um, before dying back and overwintering. And then also in Mediterranean climates like the California floristic province, geophytes are extremely diverse, where they are able to survive dormant during the hot, dry Mediterranean summers, and then take advantage of the more conditions more conducive to growth during the cool and moist winters. But once again, we don't know how differences in the development and morphology of those underground storage organs might affect the plant's ability to survive in these types of seasonal climates or different types of seasonality. So this was a question that I was interested in addressing, and I'm not going to dive into some of those statistical methods during the talk today, but I'm always happy to talk about phylogenetics and statistical modeling, so please let me know if you have any questions. Basically, what we did is we took information across the order Liliales, so that's around 1,200 species. Our data set included about half of that, so around 600 species. And we categorized plants of those 600 plants based on what type of underground storage organ they had. And then we also characterized how seasonal their climatic niche is based on seasonality of temperature as well as seasonality of precipitation. And then we wanted to ask, is there a correlation between the type of underground storage organ and the seasonality of climate? In other words, do geophytes that have these different types of underground storage organs shown here on the left, are they adapted to different climatic conditions, specifically thinking about seasonality? And exactly how we did that modeling and how we tested for that correlation, I'm sort of sweeping under the rug, but again, more than happy to answer any questions you might have about that. And in short, what we found is that geophytes that have modified roots rather than modified stems or leaves are adapted to less seasonal temperatures. We saw no significant difference whatsoever um, when we looked at precipitation. And so the sort of one major take home that we had in this project was that there's something going on differently for geophytes that have modified roots when it comes to temperature seasonality. And this convergence in the geophytic habit is not driven necessarily by temperature seasonality. Maybe it's being driven instead by precipitation seasonality. Um, but nonetheless, there appears to be a sort of special relationship between plants with modified roots and less seasonal temperature climates, which does roughly map out to be the tropics. And in fact, this picture is of a species of Bomarea, a genus that by the end of this talk you're going to know so much about, which is a tropical geophytic plant in the order Liliales. And so this is one of the things that really spurred my interest in the group Bomarea. So let's dive into Bomarea. We're going to jump from this sort of global evolutionary ecological study into a more taxonomically narrow group. And Bomarea is a genus in the family Alströmeriaceae in the order Liliales. So it is in this group of true lilies, at least at the order level. The sister genus to Bomarea is Alstroemeria, the commonly cultivated cut flower that's found in Trader Joe's. I think one of my first herbarium specimens in grad school was going down to the Trader Joe's on University Ave in Berkeley and buying some Alstroemeria, making an herbarium voucher, and taking some leaf material to have outgroups for my phylogeny. Um, but I think the Bomarea are the lesser known, but far more fascinating member of this family. This is a species, Bomarea longistyla, that's found in these really high elevation habitats, not too far of a drive from Lima in Peru. So on the drier side of the Andean Cordillera, but still quite high in elevation. There's around 120 species of Bomarea, and this is a few photos to just give you a sense of the diversity in the genus. Most species are climbing plants. So there's a nice shot right here in the middle of Bomarea tarmensis where you can see the stem coming down here. Often what these plants will do is they'll sort of weakly vine on shrubs and small trees. And then when they flower, they'll start sort of hanging downward. And so often in the forest, what you'll see is just this tight ball of flowers hanging down from the canopy above. But there's also Bomarea that are not climbers. They self-support. Bomarea suberecta has sort of a costus-like appearance. It has this spiral form. Brevis is another non-climber, and Longistyla as well. You can see there's this sort of more stiff cane-like stem as it's described. And then once the plant begins to flower, the apex recurves downward. Um, One of the other reasons that I became really interested in Bomarea is that it is extremely species rich in the tropical Andes, which is one of the most biodiverse, if not the most biodiverse region on the planet. The tropical Andes are also an iconic example of recent and rapid geological change. And I love this map and this image because by being in black and white, it really just draws your eye to the incredible topographic complexity of this part of the world. You can see how quickly you gain and lose elevation. And even in some of these regions within the Andes, how much variation there is in topography in very small areas. So I was interested in understanding more about how Bomarea evolved, how it arrived into the tropical Andes, and how the tropical Andes may have influenced its evolution, all with this context of Bomarea being a geophytic plant that had modified roots, um, and trying to understand um, for one, what a geophyte was doing in the tropics in a relative in some cases in relatively aseasonal climates to begin with. So to do this, I extracted DNA. I relied heavily on herbarium specimens for a large part of this project. Unfortunately, in my 5-year PhD, I was not able to travel the full extent of the range of Bomarea from northern Mexico down to central Chile and sample all of the species. So I relied heavily on herbarium specimens. I also did some of my own field work. And from that was able to reconstruct a phylogeny of relationships. And this on the first phylogeny, I intentionally removed the tip labels here so that we're focusing on the big picture. This was the first time that we'd had really any resolution in the phylogeny of Bomarea. Previous attempts had resulted in really poor resolution. And just very, very low variability in the primarily chloroplast markers that were used. So this was a huge improvement. I think there's still a lot of improvement to make in our understanding of Bomera relationships, both in terms of increasing the sampling and then thinking more about just being a little bit more picky about what genetic regions to look at. But we verified that Bomarea as a genus is monophyletic, that it is sister to Alstroemeria, and that those two genera shown here and here are collectively sister to this very small clade in the family called Luceriagae. This is two genera, Dromophila and Luceriaga. So then what I was interested was getting back to this question of the tropical Andes and how Bomarea arrived into that region of the world and whether this sort of intense geological process of mountain uplift and the downstream complexity that came from that influenced the evolution of this group. So I aggregated digital locality data from, again, herbarium specimens to get a sense for each species where that species occurred. And then I sorted those species into these regions that I'm showing here. So Central America and the Caribbean. I split the Andes into sort of the Northern Andes, the Central Andes. And then there aren't any Bomarea that occur in montane regions of Southern South America. So I sort of grouped Southern South America all into one. And then this region here of sort of Eastern and Central-ish South America. So what I found when I combined that digitized locality data, the phylogeny, and again, a statistical model of how we think that organisms move and disperse. If any of you were here for Ixchel's talk last month, she covered these models in detail. And what I ended up using was a simpler version of the model that she described. But what we found was that Bomarea likely originated in southern South America. And there are a few species of Bomarea that still occur in that region. One of them is Bomarea ovalle. This is a species that is found and it's endemic to the Atacama Desert in Chile. So it's extremely atypical in terms of its habitat and range for the genus. You can see here it's like it was trying to be an Andean climber and then just kind of like fell out of the mountains, landed in the desert, and just gave up and lay there. Um, it is a spectacular plant. I have not seen it in person. I would love to someday. Um, from southern South America, we found evidence for a single dispersal event from southern South America into the central Andes. So that's shown here with one arrow. Um, and the central Andes, so Peru and Bolivia, is really where the vast majority of Bomarea species are found. These are some pictures of some of the modern day species that are found in this region. It's also where there's the most morphological, and ecological diversity of species. Um, and a really just spectacular hotspot for Plumeria in Peru, Bolivia, and then also into Ecuador. Um, from the Central Andes, we found evidence for three independent dispersal events into the Northern Andes. And so what that means is that there are the current Bomarea that we find in northern South America, some examples of which are found here, are not necessarily each other's closest relatives, but instead some of these may be more closely related to some of the Central Andean species than they are to each other. Um, and we then found, and there's a good amount of diversity in the Northern Andes um, as well as the Central Andes. And from there, we found that there were again these independent dispersal events that led to the establishment of Bomeria in Central America and then north up into central Mexico. And these are some of the species that are currently found in that region. And once you get into central Mexico, there's really only two or three species, depending on who you ask. If you ask me, 2.5. Um, and what was most interesting about these dispersal events is that they really line up with the timing of major movement in Andean uplift. And so what we see is a pattern where Bomarea is really following the Andes as it rises. The mountains first started rising in southern South America, and then began to rise in the central part of the continent and then the northern part of the continent. And then you have the closure of the formation of the Isthmus of Panama and the subsequent sort of formation of mountains in that region as well. And what we see is Bomarea really tracking the availability of montane habitat during that period of intense geological change. So a couple of really major takeaways here is that we find evidence for a temperate origin and tropical dispersal. And so this is a group that originated not in the tropics, but instead in temperate conditions in southern South America. Although I will say, of course, that the climate in that part of the temperate region was quite different then as it is now. Um, that's some work that I would love to dig into a little bit more is to do some of those paleoclimatic reconstructions and see at that time what was the climate like in those regions of the world. Um, and then once the sort of Andes started rising, we see this explosion of biodiversity into these diverse biomes that are created when you have mountain uplift. These valleys that alternate between sort of wet cloud forest habitat and then dry interandean valleys, really promoting the diversification of species in this region. And we also see faster rates of diversification, so species formation in this tropical montane habitat than in the sort of northern or southern extremes of the range of the genus. And so all of this together goes to show that, at least in Bomarea, change rather than stability is responsible for the high diversity of this group in the tropical Andes. And so at these longer geological timescales, so we're talking roughly 8 million years from the sort of most recent common ancestor of Bomarea, and then 3 to 5 million years for the origin of most of the major diversity in the group. That change is responsible for diversity at these larger timescales. Even though we know that change and rapid change in sort of the current world that we're living in is causing a decrease in biodiversity overall. So this is published and available. The QR code there, if you'd like to take a look at the publication, and feel free to reach out with any if you need a copy of the PDF. I think it should be open access, but let me know if you run into any trouble. So since doing that work, I have become only more and more interested in Bomarea diversity. And part of that is because every time I go to the field, I think I go into the field with questions and I leave with even more questions. And a lot of those questions lately have been taxonomic. This is a group that is in major need um, morphological and molecular sort of combined integrated systematics. It hasn't had a monograph in the past 100 years. And so a colleague of mine, Fernando Alzate at the Universidad de Antioquia in Medellin, Colombia, are working on a taxonomic revision. And this has led to the description of some new species. So I thought I would share them here. This is Vomeria globosa, which I published with some Ecuadorian colleagues earlier this year. And this is a really interesting story because this species was, um, I think, probably first recorded to our knowledge on the Flickr account of Andreas K. Andreas unfortunately passed away before any of this work, but he had actually photographed and documented an undescribed species on Flickr. Um, the we then saw I think it was two iNaturalist observations of this same organism, um, and realized that this was an undescribed species. My colleagues in Ecuador went out to the field twice, didn't find it the first time, went back again, found it the second time, collected it, and we've described it. We then subsequently found one specimen in an herbarium and after going through the collections of Plumeria and all of the major herbaria in Quito. And this species actually occurs along a road that is quite commonly traveled and collected on. It's a place that botanists have been known to take students for sort of plant systematics field trips. So it's not an unreachable or particularly remote area, and it just goes to show to me that there's a lot of diversity in this group that has yet to be described. And I think it's adorable. This is another species that we're in the process of describing. Doesn't have a name yet. This one's from a little harder to access region of more like southeastern Ecuador. Um, and again, this is a species that my Ecuadorian colleague noticed from iNaturalist photos. And this is a species that co my colleague Fernando and I first found in the herbarium in Oxapampa, Peru. Um, we so far only know it from herbarium specimens, and I'll tell you a bit about our process of trying to document the species. So we found specimens in the herbarium in Oxapampa and in Missouri Botanical Garden herbarium. Together, there's around 6 or 7 known specimens of this plant that come from 3 total localities. All of the flowering specimens were collected in January. So we thought, okay, well, we got 3 places to go and we know when we got to go. So we planned a trip down to this region of central Peru earlier this year. Um, this is a map showing the general region. So here it's quite small, but this is Peru, and we're really just kind of smack dab in the middle. This is Oxapampa, which has a really spectacular herbarium for a pretty small town that's um, works very closely with the Missouri Botanic Garden to document the diversity in this area. And there's a national park in the region as well. The localities were approximately in these 3 places. Um, and it doesn't look, it's hard to get a sense of scale here, but it took us about like a full day driving to get to Iscosacin, and then from Iscosacin, a half day driving to get roughly to this point. We then walked for the full rest of the day that we had and couldn't find any individuals in this area. This area would have been another like 1 or 2 days by foot, or um, I guess we could have tried to use horses. We didn't find it here, so we went back to Oxapampa and then we made one last push to try to get to this point, which you can drive here. It's about a 2 or 3 hour drive, and then walk in for about 4 to 5 hours to this point, and didn't find it there either. So this is my colleague Fernando trying to sort of cross this river by sliding over this fallen log to search for it in a couple of those areas. Um, and this is a lot of what the region looked like. This is an area that is, it's people live there, people are trying to survive in this region. And so there is a fair amount of cattle raising, and there's also the forests are fairly fragmented, so you can really see here the impact of that in creating a pretty patchy landscape for the remaining intact forest. Once you get into those patches, it's beautiful. It's this much more lowland habitat than most Bomerias like. And again, in those two places that we were able to get to, we just saw no like not even a single vegetative shoot or like dead leaf. And we've both at this point got pretty good eyes. So if it were in those places that we searched, we would have seen it. Um, but it was not all for nothing. We did see some other really beautiful Bomeria, including we'd been in that region before, and these are two species that we hadn't collected previously. So nonetheless, a good trip. Um, and I want to also just take some time now to talk about sort of, so I've got this like taxonomy stuff that I'm working on, which is really exciting. And this other direction that I'm really interested in moving with Blomeria is thinking about the biotic factors that may have influenced its diversification. So I told you the story about the impact of Andean uplift, dispersal on diversification of the group. That's not the whole story. The sort of diversification is such a complex process and is almost certainly influenced by all of these different factors that the plants are experiencing, their abiotic environment, as well as their biotic interactions. And so to start thinking about these more biotic interactions side of the story. In March, my research tech, Chiara, and I went to Bogota in Colombia at the National University. We had this great team, Felipe Mesa, who's an undergrad at UNAL, and Jonathan, who's a tech with a hummingbird research station, to start thinking about what are some of the primary pollinators of Bomarea, and how might those interactions be shaping the evolution of this group. Um, and we knew going into this that most Bomarea are thought to be hummingbird pollinated. I mean, they just scream hummingbird with like large, red, showy flowers. Many of them have produce exorbitant amount of nectar. And hummingbirds have been sort of colloquially observed to visit the flowers. I've seen them, but there's not been much of a concerted, systematic effort to see whether certain species are pollinated by specific species of hummingbirds, whether like to what degree is this pollination more of a generalist strategy. So do we also see insect visitors, or is it strictly birds? Um, and how might those pollinator visitors be impacting both sort of gene flow between species and then potentially the process of speciation? But the first step was really to gather basic natural history information. And so we went to Chingasa National Park, which is close to Bogota in Paramo habitat. So this is high elevation, tropical, alpine, generally very moist habitat, famous for Aspiletias, these fantastic members of Asteraceae that often dominate these ecosystems. And there's 5 species of Bomarea that grow here. There's a great field station, great team of parks rangers. So we had sort of the best setup possible for trying to film plant pollinator interactions and gather preliminary natural history data about Bomarea, thinking about biotic interactions. So we brought cameras into the field and we set up tripods to spy on flowers. We'd leave the cameras for 4 to 6 hours, ideally, and then look back at the footage and see if we had filmed any interactions. Um, we were in a national park where there are visitors, so we had to do a really good job of hiding the cameras. So you can see here that we got a like camouflage poncho and camouflage netting. And then we would just pile vegetation around the camera. So you can see here we put this true scale around the camera so that anyone driving by on the road wouldn't just like see expensive camera gear, stop and take it away. I think we did a pretty good job. They were a little too hard to find sometimes when we'd go back to get the cameras after the 6 hours had passed. And then after filming, we would bag the inflorescences overnight. And the next day, we would measure nectar concentration. We tried to measure approximate nectar volume. Getting exact nectar volume measurements was not super feasible. But looking at nectar concentration, so the amount of sugar in the nectar, which can be a really good indicator of or it's often tightly associated with bird versus insect pollination. Um, and then did a whole bunch of other measurements looking at floral characters. And this is all pretty preliminary stuff. My student research tech Chiara is going to be talking about this at a talk at the botany conference. So if you're there, be sure to check out her talk. But here's some videos of what we captured. So in this case, we've got a hummingbird. It's visiting this Bomarea hirsuta, and I'd say that that bill is just maybe like a little too long to be a good fit with that flower, right? Like the anthers inside that flower are not coming into contact with the bird's head. Pollen can probably adhere to the bill, but not nearly as well, presumably, as to the feathers. And then we have an example like this, where this hummingbird, a little sneaky guy, isn't even pollinating this plant at all. Its bill is too short to reach the nectaries. And so it's taking advantage of these nectar robbing holes in the tepals of the flower. You can see the holes here, here, here. And almost all of the flowers have these little nectar robbing holes. And so this hummingbird with its short bill is taking nectar without coming into contact whatsoever with the plant's reproductive structures. So the first one was too long, this one's too short. And then we have this example of just right. This is a really fast video, so I slowed it down to 25% speed, but you can see here that that bird's bill is just the right length so that that bird's head is coming into contact with the anthers in those flowers. Okay, so that finally brings us at the end of the talk to the Pacific Northwest. Like I said, I've been here since September of last year, so just under a year. Um, but I'm excited to be in this area and to be learning the flora of the Pacific Northwest and thinking about how those questions that I'm interested in can be addressed by lineages that are diverse in this region. And so one group that I want to touch on just very briefly. And I should say that these are geophytes, right? Like we love geophytes. We're sticking with the geophytes, and there are some really spectacular geophytes in the Pacific Northwest. These are just some examples of them. We've got some members of the sort of true lilies and Liliaceae. And then I'm going to tell you a bit about these two lineages here. So first, onions, allium, fantastic group. We've got around 22 or so species in Washington. They're super diverse in California as well. And my PhD student, Emily Humphreys, is starting to work on Ameralium, building off of some previous work that was done based in California with New Nguyen, Chelsea Speck back in 2008, looking at the sort of systematics of this group and really just adding to the taxonomic sampling, filling in some clades that had lower representation, and then expanding the set of molecular markers that we use to reconstruct those phylogenies. And then thinking about the evolution of stress tolerance, specifically ability to grow on stressful substrates like serpentine soils, but not just serpentine, also thinking about gypsous soils and other inhospitable to most plants, or substrates that are inhospitable to most plants. So Emily will be giving a poster at the botany conference. Definitely check that out if you have an interest in allium and and or soil endemism and soil tolerance. And Emily was just down in California until a couple of days ago, and shared this spectacular picture of allium somewhere from the great state of California. Um, Okay, and then the last, this is really where we just get into like wild speculation stage of the talk at the very end. Um, but one of the many deep dives that I've gone on while learning the flora of the Pacific Northwest this past year is reading up on Ulsinium. So Ulsinium is sister genus to Sisyrinchium in Iridaceae. It's much smaller in terms of species number than Ulsinium, quite morphologically similar as well. So should Ulsinium just be considered part of Sisyrinchium? Maybe. I don't, not here to argue about sort of the taxonomic decisions there, but instead to point out that one thing that the split between Sisyrinchium and Ulsinium did was maybe draw more attention to the fact that there are plenty of Sisyrinchium species that are found throughout the Americas. It's a maybe not continuous distribution. There's certainly less diversity in the more tropical regions, but we have many members of Sisyrinchium that make it throughout the west coast of North America and are quite diverse in northern South America, and there's members in Central America as well. Ulsinium, on the other hand, is restricted to southern South America. The northernmost member of this group gets into central Peru, with the one exception of Ulsinium douglasii, which is common. It does make it down into California, Northern California, but quite diverse or quite dominant or common in Oregon and Washington, and then somewhat farther north as well. There hasn't been a huge amount of work on this genus because it is kind of like the lesser cousin of Sisyrinchium. So a study from 2017 that included some members of this group found that the PNW species douglasii is actually sister to a species that is currently restricted to the Falkland Islands in southern South America. And I just think that this just disjunction is really quite fascinating. It implies that there was a dispersal from southern South America to the Pacific Northwest, most likely, which, given the sort of general pattern of disjunctions, is not the most common pattern that we tend to see. So, this is another sort of interesting group with it's got some South American stuff going on, it's got some Pacific Northwest stuff going on, interesting biogeography, underground storage organs, monocot, checks all the boxes. So these are the types of things that I'm hoping to pursue. Think moving forward in my career here, very excited to be back on the West Coast and be working with other Western North American botanists. So with that, I'll say thank you to all of the people that made this talk possible, collaborators on projects, past, present, and future. And thank you all for being here. My email is on the slide. Please feel free to reach out. And of course, happy to chat now.
Thank you so much, Carrie, for that amazing talk. So we already have a few questions on the chat. If other people have questions, please feel free to raise your hand or put it on the chat, and I'm gonna read them. So I'm gonna do one from the chat, and then whoever has their hand up, right? So the first one, actually, Andy Simpson, you can just unmute yourself. You had a question on the chat, so you can just probably ask it.
And do you think
Andy?
good? Oh, you got to let them unmute.
There should be able now.
There we go. Um, so the first question I want to ask is, can you give a time frame over which the different regions of the Andes uplifted and resulted in, and you are hypothesizing resulted in Bavaria expanding northward as a consequence?
Yeah, I don't have the exact dates of the various stages memorized. There's a great paper by Karina Horn, I think it's 2010, that has this awesome figure showing the different sort of what the South America looked like at these different periods of time. really, so Southern South America started uplifting earliest. Maybe like that's like Miocene or
Miocene.
so
Okay.
like 15-ish perhaps, and then but you don't start getting um with the Bomeri connection, it was um really linked with the final stages of uplift in those particular regions, not the onset of uplift. So the Andean orogeny process started before we see Bomeria starting to diversify really around 5 to 3 million years. But the Andean uplift started for millions of years before that. It's really once you start getting to the final stages where you have sort of higher elevation ecosystems in those temperate zones.
So
Sorry, I can't provide a more specific answer, but I'm happy to send you an
okay.
email
So,
later.
so
Yeah.
it's not possible. It's not possible then that that we're looking at a migration from Antarctica and the Eocene.
Oh, this is such a great question because what I didn't tell you is if you look at the phylogeny of, and this we suspected prior to my work, I'm not going to take credit for this, um, but if you look at the phylogeny of Alstroemeriaceae, Luceriaga and Dromophila are found in like that clade is found in southern South America, New Zealand, and Australia. And so that is
Okay.
like, this clear. Yeah, so if you go deeper in the tree, you totally see that pattern. It's so neat. I love it.
Okay. Neat.
Thank you so much, Peter.
Oh, I don't know that I've ever seen a talk that made systematic seem like such a grand international adventure. Um, I was wondering if you could see any ways in which geophytic habit played a role in the diversification of Bomeria.
Yeah, this is what keeps me up at night, Peter. Then this is, I sort of mentioned offhand that I would love to do some paleoclimatic reconstructions to get a sense of like what was the climate like in southern South America when these lineages, both Bommeria and Alstroemeria has tuberous roots as well, just a little bit less extreme, I'd say. Um, when those two lineages started diversifying. And Alstroemeria tends to stick to more seasonal climates in terms of precipitation. They're super diverse in Chile and that Mediterranean climate, and then up into Brazil and some of the seasonal climates in Brazil as well. With Bommeria, um, in what one thing that I suspect this is very there's no data here, this is just observational. But in parts of the central Andes, there's such topographic complexity that you have species that are growing in close proximity distance wise, but very different climatic zones. And so there are these like pretty dry valleys in parts of the Andes. And plants that grow in those dry valleys flower at a completely different time of year than plants that are growing like, a 1.5 hour drive over into these like very moist cloud forest conditions. And so one thing I suspect is having this geophytic habit maybe permitted species to move into a wider breadth of climatic conditions. And so it was sort of like a precursor state that then enabled subsequent diversification into a wider range of climatic conditions and enabled them to take advantage of the breadth of niches that opened up during uplift. But that's again very um, that's just an idea, no actual data to back that up at this point.
Thanks.
Yeah.
Thank you. Any other questions that you want to ask directly? You can raise your hand and I'll call you. Otherwise, there's a couple on the chat. The first one is, do the underground parts produce exudates during dormant seasons, potentially sustaining a subsurface microbiome?
This is such a great question. I don't know. I you so I gave I spoke about some of this work earlier about a month ago, and there was a member of the audience who mentioned a study that I should have followed up on and didn't, that compared microbiome composition between geophytic versus non-geophytic plants in like sagebrush, I think it was. and did find compositional differences. And so I think that there's evidence to which this makes total sense to me, like that there would be microbiome differences between geophytic versus non-geophytic plants. But my sense is that this is also a relatively underexplored area, and super interesting because plants that invest so much energy and resources in these underground storage organs, that's their whole life savings down there for the next growth period. And so thinking about microbiome defense and just in general, or so microbes as defense, but also as potential herbivores, and then anti-herbivory mechanisms beyond the microbiome, I think is super interesting. It's one of the things that got me interested in studying geophytes, and I've never really followed up on. It's a great question.
Thank you. There's one more. So speaking of allium and Bommeria, and if you consider that genera of geophytes from the Andes and in the Pacific Northwest have a history of indigenous use, have you noticed any morphological adaptations or migration in response to human or animal interactions? And also a follow-up question on that, would these explain some disjunct population?
Yeah, it's such a great question. So, I'll talk about Bommeria first. There's one species of Bommeria, um, Bommeria edulis, that has the most documented use by indigenous communities, mostly prior to European conquest of the Americas. It's mentioned in the species description of this species. Of the sort of species description is from modern day Haiti, and it describes people in Haiti um, gathering Bommeria tubers from the wild, eating them. And it even includes kind of like a rough recipe for how to extract starch from these tubers and cook it. So, and that species has by far the widest distribution of Bommeria. It occurs from central Mexico all the way down through Central America, northern South America, east into sort of sticking mostly to the coastal areas of Brazil, down into northern Argentina. Um, and uh, I do believe that extensive distribution, it's not disjunct, but it is quite large, is likely due to human mediated dispersal or moving around this valuable plant. Um, the PNW side, I don't, so allium certainly used by indigenous peoples still today. One thing about these bulbous plants is that they often will produce bulblets, or little sort of clonal propagules. Well, the bulbs will basically branch, and so if any organism, human or other, is digging them up, um, you like if I were harvesting allium, I would only take the large onions, right? The small ones just sort of fall. And so that is, I think, a mechanism for living in for the plant to be living in these environments where it's being dug up, it's being potentially consumed, and then still having these asexual propules that survive and continue on. Um, I would say that I do not know enough about like allium distributions to hypothesize about human like movement of those plants shaping distributions of allium. I just don't know enough about the lineage, but you could ask Emily that question at Emily's poster if you're at botany. Get ready, Emily.
All right, thank you very much. Do we have any other questions?
I just wanted to connect you potentially with someone who's doing research that might interest you that you may not have heard of. So Curtis Lubbe, Lubbe, who's now at the Institute of Botany at the Czech Academy of Sciences, has been putting together a sort of encyclopedia of all of the various bulb forms in Oxalis. There's got 100 species,
Oh,
I think,
wow. Yeah,
now.
Oxalis is spectacular.
And there's a lot of variation. And I don't know what all he gave a talk on it, and I don't know what all it's is published yet, but that might be a fun person for you to chat with. So it's Lubbe Curtis, first name, and he's at the Czech Academy of Sciences.
Fantastic. Thank you so much for that. Ish, there is a question that came just as a direct message to me, so I can
Okay.
read it and answer it. Um, do you see speciation or diversity increases with more recent events like deglaciation? So with Bomeria, there were only a couple of examples that I could find where there were sister species that occurred sympatrically, roughly, or like in the same general region that had split around the time where we saw these glaciation and deglaciation cycles. And so it's not, I would say it's not very common, and that didn't seem to be a primary driver of speciation in this group. What I do think might be happening is that in Bomarea there are far too many species complexes. So these just taxonomic messes where there's all this morphological variation. We can't seem to figure out how to sort it into these neat units that we could bin into species. Um, and several of those complexes, some of the most prominent ones, are restricted to high alpine conditions in the tropics. And so I do think that perhaps not speciation per se, but certainly in the generation of diversity that has led to these species complexes that occur in conditions that were, or regions that were likely um, connected and disconnected repeatedly due to glaciation cycles.
Awesome. All right. I don't see any more questions. So with that, I really want to thank you, Carrie. That was a fantastic talk. And thank you everybody for joining us tonight. Um, we'll see you in a month from now with the talk of our next speaker.
Thank you.